Hello. Before we continue with our discussion of hormones, I would like to remind you that quiz 4 is due on Sunday night at 11:30 on Moodle. It's closed book, so please put your books and notes and other information away when you're taking the quiz. Do it on your computer or I suppose on your phone and turn it in. The The content of the quiz is hormones, the organs that secrete hormones, the names of those organs that secrete specific hormones, and the names of the organs or cells that respond to specific hormones. So learn about hormones, their sources and their targets, and learn more about the muscles of the legs, because that is covered on the quiz as well. Okay, so today we are going to talk about some more about growth and metabolism. We will focus on thyroid hormone and growth hormone. And then we will talk a little bit about homeostasis, endocrine homeostasis of water and electrolyte levels. The control of salts and water is really covered in AMP2 when we cover the kidney, because that's the most important organ for it. We discuss the mechanisms of how these hormones work on the kidney in AMP2, but I can't let you get away from the endocrinology of this here without talking about vasopressin and parathyroid hormone and calcitonin here. So we will not talk about mechanism in the kidney because that's AMP2, but we will talk about endocrinology and the endocrine control of salt and water balance. And then next week, starting on Tuesday, we will talk about male and female reproductive endocrinology. So this is coming up next week. All right, so we're talking about growth and metabolism still, and now let's focus on thyroid hormone. So the thyroid hormones, T3 and T4, are iodinated derivatives of tyrosine. Here is their structure right here. This is the structure of T3 because it has 1, 2, 3 iodides attached to the ring structure. T4 has an iodide here. That's the only difference between T3 and T4. You should be able to recognize the source here, that this looks like tyrosine, and that is the source of the T3 and T4 hormones. That's the amino acid that is modified to make T3 and T4. So T3 and T4 are iodinated tyrosine derivatives. The stimulus for the production and secretion of thyroid hormones is TSH, thyroid stimulating hormone, from the thyrotrophs of the anterior pituitary. They, in turn, going back upstream, are stimulated by TRH, thyrotropin releasing hormone, from the hypothalamus into the median eminence. The cells that synthesize and release T3 and T4 are called the follicle cells of the thyroid gland. We'll talk next about the synthesis because it's an interesting path. The targets of thyroid hormone are many cells: liver, kidney, heart, the nerves of the central nervous system, brain, muscle cells. So many, maybe most cells in your body respond to thyroid hormone, and they respond because they express an intracellular receptor for thyroid hormones that causes changes in gene transcription in those cells. And this is a list of only some of the effects in those cells. It increases the rate of cellular respiration, so it makes more ATP. It increases the metabolic rate, so everything that the cell does, it does more of. There is the increase in ATP production, increase in sodium potassium ATP ACE activity, production of heat by metabolism, and the development of cells, development of the body, somatic development, especially in the nervous system. So the development of a functional nervous system in humans really depends on the presence of thyroid hormones during neural development. The thyroid gland, that's just shown here, the thyroid gland is located in the throat, wrapping around the trachea. Let's talk about the synthesis of T3 and T4. So this is a slide from your textbook. Let's get oriented here. So here is blood flow, a capillary of blood serving the thyroid gland. This layer here is supposed to illustrate layers of cells. These are cells of the thyroid that line a lumen called the colloid. So we have blood flow over here, blood flow, and cells of the thyroid. The cells of the thyroid produce a protein called thyroglobulin. Thyroglobulin is an important player in this story. Thyroglobulin is a protein, and it's produced in the follicle cells. So thyroglobulin is produced in the follicle cells, and then it is secreted into the colloid, the lumen of the follicle, the colloid of the follicle. So here is a cartoon of thyroglobulin in the lumen in the colloid. Iodide, which is an essential nutrient that you get from food, is transported into follicle cells by a sodium iodide co-transporter. So now we're getting all the way back to the beginning of the course, and we're learning about symport of sodium and iodide into the cells. So you get iodide from your diet, your body absorbs it in the intestines, it's in blood, and then the cells of the thyroid specifically express this iodide sodium co-transporter to pump iodide into the follicle cells using the sodium electrochemical gradient, as we've talked about at the beginning of the course. Iodide diffuses, and it in fact diffuses through the internal membrane of the follicle cell. In the lumen of the follicle, there are enzymes that iodinate tyrosines of thyroglobulin. So here, iodide that has been transported into the follicle and into the lumen, that is to say, into the colloid, is oxidized and attached to the rings of the tyrosine. This is done by enzymes in the colloid. And so you can see here is a tyrosine that has become monoiodinated tyrosine. And here's a tyrosine that has become diiodinated tyrosine. So there are enzymes that attach iodide to the ring structure of tyrosines on thyroglobulin. There is another enzyme that translocates one of these iodinated rings onto another. So it takes one of these iodinated rings and attaches it to another. and makes the what is going to become the thyroid hormone with the two ring structures. And if a two ring iodine, I mean, if a monoiodinated tyrosine is attached to a diiodinated tyrosine, it becomes T3. The mono is attached to the di, it becomes T3. And if two diiodinated tyrosines are attached, it becomes T4. 1, 2, 3, 4 iodides. So now we have an iodinated, we have tyrosines that have become modified by getting two of the ring structures attached and a number, 3 or 4 iodides attached. This is in the colloid, and this thyroglobulin, this modified thyroglobulin, is then endocytosed back into the follicle cell. So here is endocytosis, clathrin coating, all that good stuff you learned about in cell physiology about endocytosis. Here is the modified thyroglobulin in a vesicle of the follicle cell, and lysosomal enzymes. So this is really a lysosome, not just a vesicle, it's a lysosome. Enzymes hydrolyze the ring structure, the double ring structure from thyroglobulin to release the T3 and T4 from the thyroglobulin. T3 and T4 are amphipathic enough to diffuse through the membrane into the bloodstream, where they travel to all parts of the body and interact with most cells in the body to maintain a healthy level of growth and metabolism. The thyroglobulin is recycled, and production of the production of the T3 and T4 continues. This whole process is stimulated. Everything about this process is stimulated by TSH. Okay, here's a fill in the blank slide to review the control of the production of thyroid hormone. So what goes in these blanks? What controls the production of thyroid hormone? Give you a few seconds to do this, and then we'll look at the feed forward and feedback control of thyroid hormone production. All right, so I hope you remember that the hypothalamus releases thyrotropin releasing hormone, TRH, into the median eminence. It flows through the portal circulation. It interacts with TRH receptors on the thyrotrophs of the anterior pituitary gland that releases thyroid stimulating hormone into the systemic blood. The systemic blood goes everywhere, but the targets, the receptors for TSH are expressed on the follicle cell of the thyroid gland, which are stimulated to proliferate and to produce T3 and T4. So TRH stimulates the release of TSH, which stimulates the proliferation of follicle cells in the thyroid gland and stimulates them to secrete T3 and T4. TRH is a peptide hormone, TSH is a peptide hormone, and T3 and T4 are iodinated tyrosine derivatives. TSH does negative feedback, and T3 and T4 does negative feedback to maintain homeostasis of T3 and T4 production. Let's go on to look at the next slide where we will talk about pleiotropic, that is to say many effects, the many effects of T3 and T4. Oh, before I go on, let me just mention that T3 and T4, being amphipathic, are not stored in vesicles and released upon command by activation of TSH receptors. It's the synthesis of T3 and T4 that is activated by TSH. Because once it's synthesized, T3 and T4 will diffuse out of the cell into the blood. So what are the effects of T3 and T4? Well, many. Growth of cells, control of their metabolic rate, control of their ATPase activity and heat production, cell division that you learned about in CGI, cell development, that is to say specialization of cell type that we don't really cover because we don't have a developmental biology class that would do this in detail, but development of cells is very important. And in this case, T3 and T4 are especially important for the development of neurons in the brain and in the central nervous system. So let's use the knowledge of thyroid endocrinology and talk about some of the abnormalities of thyroid in the thyroid endocrinology. So hypothyroidism, low, low production of thyroid is a problem. It's a medical problem. What would we call primary hypothyroidism? Well, primary hypothyroidism means that the thyroid gland is the source of the problem. We don't have a problem in the hypothalamus. We don't have a problem in the anterior pituitary. We have a problem in the thyroid gland. That's what makes it primary. Secondary hypothyroidism might be a problem with the production of TRH or the production of TSH. That would be secondary. The causes of primary hypothyroidism there are two major causes. One is not getting enough iodide in the diet. If you have a diet that is deficient in iodide, then your thyroid glands can't produce thyroid hormone. And there might be nothing wrong with your cells, but if they don't have the iodide, they can't make thyroid hormone. And that is why iodide is often supplemented in table salt. You may, if you've read the label of, Morton's table salt, many of the canisters of it will say that it is iodized. And that means that there is a small amount of sodium iodide mixed in with the large amount of sodium chloride in order to provide a small amount of iodide in the diet when you salt your food. Another defect that causes primary hypothyroidism is an autoimmune disease called Hashimoto's disease, where the immune system somehow starts to recognize the follicle cells of the thyroid as not part of your body and attacks it and causes autoimmune destruction of the follicle cells of the thyroid. So let's just think about iodide deficiency, because that's the most common form of primary hypothyroidism. What are some of the symptoms of primary hypothyroidism? Well, think about what would happen if you didn't have enough of the thyroid hormone that was stimulating metabolism and growth and things like that. So low metabolism can lead to a gain in weight because your appetite might not be changed, but your metabolism is low, and so weight is gained. The calories you eat goes into fat. Fatigue, low metabolism, a sense of weakness and fatigue. Remember that thyroid hormone sustains ATPase activity and the somatic, the cellular production of heat. And so people who are hypothyroid generally are intolerant to cold temperatures. They're the people who are wrapped up inside in, sweaters and scarves and things because their body is not producing enough heat. Bradycardia. Brady means slow, cardia means heart. So a slow heart rate is often a symptom of primary hypothyroidism. And now here is a problem that I want you to think about, and I'll pause the slide after I talk about hyperthyroidism. Goiter is a visible symptom of primary hypothyroidism, especially caused by iodide deficiency. It's not autoimmune destruction of the thyroid. Goiter is a enlargement of the thyroid gland, and sometimes it can be quite extreme. And so the question I want you to think about is, why would there be goiter in iodide deficiency primary hypothyroidism? Think about that for a moment. I'll pause the slide after we get to hyperthyroidism. So hyperthyroid, too much thyroid hormone, can be caused by a tumor of the thyroid gland. It can be caused by an autoimmune disease, but this is an interesting one. This is Graves' disease. In Graves' disease, the immune system produces an antibody that activates the TSH receptor. And so this is an immune activation of the thyroid. And so it hyperactivates the thyroid and causes hyperproduction of thyroid or hyperthyroidism. Infection of the thyroid gland can sometimes cause proliferation of the cells and activation of the cells and hyperthyroidism. The symptoms of hyperthyroidism are tachycardia. Tachy means fast, so this is a rapid heart rate, anxiety, weight loss because the cell metabolism is hyperactivated, and so your body is burning a lot of calories. Heat intolerance, your body is producing a lot of heat, and so these are people who are just they're just intolerant of any warm temperatures. Goiter. Now, in this case, goiter should be obvious if there's a tumor or if there is an autoimmune activation of the thyroid gland or a swelling of the thyroid gland that can cause a goiter, a swelling and enlargement of the thyroid. So that one is obvious. And there's an interesting phenomenon called exophthalmia. The eyes seem to bulge out of the sockets in hyperthyroid patients. This is due to swelling of the muscles and the fluid behind the eyes, sort of pushing the eyeballs out of the skull. So now I want to pause the slide and ask you, in the case of the goiter here, it's pretty obvious. Activation of the cells by a tumor or by an autoimmune, by a Graves' disease will cause the thyroid gland to enlarge and cause a tumor. Why would iodide deficiency also cause goiter? The answer to my question about why primary hypothyroidism would, especially due to, say, a dietary deficiency in iodide, would cause goiter is the lack or insufficient negative feedback. So remember, we need iodide to produce T3 and T4. If there is inadequate iodide, then we don't make enough T3 and T4. The lack of T3 and T4 interrupts the normal negative feedback to the hypothalamus and to the producing TRH, and to the pituitary gland producing TSH. So there is inadequate negative feedback leading to excess production of TRH and excess production of TSH. Those cause excess stimulation of the thyroid to produce T3 and T4. The follicle cells of the thyroid are being stimulated because they're being stimulated by TSH, excess TSH, excess proliferation of the thyroid gland trying to make more T3 and T4, but it can't because there is not enough iodide. So the thyroid gland is stimulated by TSH to make more T3 and T4, but it can't. But the cells can still develop and divide and grow and swell. And so we get an enlargement of the thyroid trying to make T3 and T4, but it can't. So the answer is, in the hypothyroid situation, there is inadequate negative feedback, excess TRH and TSH, excess stimulation of the thyroid cells, excess proliferation of the cells, and an enlarged thyroid gland in primary hypothyroidism. All right, now let's turn our attention to another important hormone for the control of growth and metabolism, and that is growth hormone. Let's review what growth hormone is. It's a protein. It's a small protein of 191 amino acids. I won't ask you to memorize that detail. It's a small protein. The stimulus for production of growth hormone by the somatotrophs, so it's produced by the somatotrophs of the anterior pituitary, and the stimulus for production of growth hormone by the somatotrophs is GHRH, which is released from the hypothalamus into the median eminence. And so the somatotrophs are specialized in the sense that they express the GHRH receptor and they produce GH. GHRH stimulates the somatotrophs to produce growth hormone, and somatostatin from the hypothalamus inhibits the production of growth hormone. So this is another classic example. GHRH and somatostatin are another classic example of antagonistic control systems to control the level of growth hormone. GHRH stimulates growth hormone and somatostatin inhibits growth hormone production, and the combination then finds a balance for normal growth hormone production. The target cells for growth hormone are many. The liver, we'll talk about IGF-1 in a minute, the liver, muscle, fat, and bone. So body size, let's talk about body size for a minute, because that's what you think of when you think of growth. Body size is primarily determined by the length of your bones. Now, we don't talk about bones in this course very much at all, and some physiologists would say that that's a mistake. So let me just remedy that mistake very slightly by mentioning that bone is an organ that is important, and there are cells, osteoblasts, chondrocytes, and osteoclasts that are important for the production of bone. Chondrocytes primarily make up the cartilage and the collagen of bone. Osteoblasts are the cells that lay down the mineral, primarily calcium phosphate and calcium apatite, the mineral bone. Osteoblasts produce mineral bone. Osteoclasts break down mineral bone. There is a balance between osteoclast activity breaking down bone and osteoblast activity building bone. And so the production of bone is a dynamic process that goes on throughout your life. During growth, childhood up to puberty, the osteoblasts are laying down new bone and making the bones grow longer in length. Osteoclasts are still working, but they are not working as fast, and so bones grow, and your body grows in response to bone growth. At puberty, at puberty, a structure forms at the end of your long bones, at the end of the bones of your arms and your legs and the rest of your body. Normally at puberty, the bones develop a plate at the end that prevents further osteoblast growth. And so the bones stop growing in length at puberty. And this is due to the increase in the reproductive hormones. That doesn't mean your bones stop doing things. Your bones still, even though they are no longer growing, the osteoblasts and the osteoclasts are still working, producing bone and breaking bone down. So if you break a bone during adulthood, the healing of bone is due to the osteoblasts putting down new mineral at the site of the injury, and the osteoclasts breaking it down, trying to help the bone heal. Breaking it down being, clean up the site of the injury so that the new bone that the osteoblasts are laying down has a better substrate for integrating into the actual bone. Okay, bone. The receptor for growth hormone is a receptor tyrosine kinase. And so this is the story of dimerization, cross-phosphorylation, recruitment of adapter proteins, recruitment of RAS, and the activation of a MAP kinase cascade that leads to gene transcription regulation and metabolism, as well as proliferation of the cells. So the effects of growth hormone, right here, as we've been talking about, increasing the number of cells, especially bone, but increasing the cell numbers of muscle cells to help move that bone, and liver cells. And so there's just an increase in cell number in a response to growth hormone. There's an increase in fatty acid metabolism. So you burn fat in order to produce the glucose that you need for all that growth. Increased in gluconeogenesis, increased in glucose synthesis, decrease in glucose from the liver so that the glucose is diverted to the cells that are growing. In the liver, growth hormone's activation is synergistic with another factor called insulin-like growth factor. The I in IGF-1 stands for insulin. It's not insulin, it's insulin-like. Insulin-like growth factor is a required synergistic molecule for growth. So it's not just growth hormone that leads to the development of bones and muscle. It's growth hormone and IGF-1. IGF-1 is produced in the liver to help growth hormone, produced in the anterior pituitary, control body size. There is a diurnal rhythm, a daily rhythm in growth hormone production. So this is data showing growth hormone as a function of time of day. So during the day, growth hormone levels are low. There's some slight oscillations, but it's generally low. During the night, when you are sleeping, should be sleeping, at least some of the night, there are spikes in growth hormone production during the night. And especially in childhood, where growth is really important, the requirements for sleep lead to production in growth hormone, and that means that sleep is an important part of growth. And so, if your grandparents make some exclamation, when you were younger, you might have heard your grandparents say, "Oh my God, it looks like you grew overnight." Well, of course, they didn't detect somatic growth overnight, but in fact, growth hormone production at night during sleep is important for body growth, especially during childhood. As I said, body size and somatic cell proliferation is something that is controlled. We have a sort of set point for body size and for cell proliferation. We're not all set at the same size. There's some variation, human variation in body size, but there is a set range of body size and of cell proliferation. And growth hormone controls that. So now let's think about what goes in this box and what goes in this box. What hormone would be higher if there was low somatic cell proliferation in order to try to bring the cell proliferation back towards a set point? And what hormone would be produced if there was excess cell proliferation? What hormone would be produced to try to reduce cellular proliferation and prevent giantism, to prevent the cells and the body from getting too large and bringing it back towards the set point? I'll pause here. You should be thinking about a hormone that goes in this box and a hormone that goes in this box that work against each other to control body size within a certain range. The answers are growth hormone and somatostatin. If there is low somatic cell proliferation, growth hormone is produced at a higher level than somatostatin, and the body's cellular proliferation is increased. If there is excess somatic cell proliferation, somatostatin is produced, which inhibits growth hormone. It inhibits the production of growth hormone. And then, so somatostatin, well, really, so this should be growth hormone, and over here we should have growth hormone and GHRH, because GHRH stimulates growth hormone, which stimulates growth, and somatostatin inhibits growth hormone, which inhibits growth. So your answer, if you had said GHRH over here, that would have been correct too. So if we were in class, we would be having a discussion about growth hormone, just as a way of activating, activating classroom. Here are two scenarios. A child is small. He is below the 5th percentile in height, but he's otherwise normal, no other abnormalities. But his parents want to give him growth hormone to help him grow bigger. They know that being too small is not an excellent place to be in our society. Many people prefer to be on the tall side than on the short side. Well, is this a good idea? Is it a good idea to just to inject growth hormone in order to increase growth of an otherwise normal child? So that's a debatable point. And what test would you want to run before you started just injecting growth hormone into a child? Instead of discussing this here, I think what I'll do is I'll post this to the forum and we can have a discussion about it. And I'll give you an opportunity to answer these questions. Is it a good idea? What tests would you run before you started growth hormone activity? And you could even say, what do you mean short stature is a problem? I'm short, I'm doing fine. So that would be an interesting debate we could have on the forum. And you can be anonymous if you want. Here's another case. A child has a small stature and in this case has severe cognitive deficits. His parents have read about growth hormone and they say, let's give him growth hormone. That'll make everything better. Again, is this a good idea? What tests would you want to run before you started growth hormone therapy? I will post this to the forum and we can have a debate about it. Let's do a clicker related to growth hormone. Why is it more difficult to measure GHRH than it is to measure GH? So read the answers. I'm going to pause this slide now so that you can look at these answers, come up with your answers, and then click forward. All right, why is it more difficult to measure GHRH than it is to measure GH? Well, B and D are the correct answers. GH concentration in systemic blood is higher than GHRH concentrations in systemic blood, making it easier to measure. Remember, growth hormone is produced by the somatotrophs of the anterior pituitary, and it goes throughout the body and it has effects throughout the body. GHRH is produced by the hypothalamic neurons. It is released into the portal circulation. Its target is really right there in the anterior pituitary, or very close to the median eminence where it is released. And so its concentration in portal blood is high, but its concentration in the large volume of systemic blood is low. In fact, very hard to measure. It's so low. And that is answer D here. GH concentration in portal circulation is high, and it would be detectable if we could sample the portal circulation, but it's so tiny that's hard to do. GHRH concentration in the large systemic circulation is low. It's diluted. Let's look at the ones that are not right. Growth hormone is a protein, but GHRH is a steroid. No, that's not correct. GHRH is a protein also. GHRH receptors are expressed on a wide variety of cell types. Well, that's a true statement. That is a true statement, but it doesn't have anything to do with the difficulty to measure it. Difficulty to measure it has to do with the concentration in blood, not the receptors. So this is a trick question because this is a true statement, but it is not the answer to the question. All right, now let's turn our attention to the control of water and salt balance in the body. We have a narrow range of acceptable concentrations of water and salt. Sodium, potassium, and calcium are critical electrolytes that we must control in our body, control the levels of, and we have hormones that are essential for that. Vasopressin, which is also known as antidiuretic hormone, and aldosterone are important for control of salt and water. We're not going to talk about atrial natriuretic peptide, but we are going to talk about vasopressin, which is also known as antidiuretic hormone, and aldosterone. It's not listed here, but aldosterone is another important hormone. And we will talk in the control of calcium levels. We will talk about parathyroid hormone and calcitonin. So that's where we're going. Let's move on. All right. Vasopressin, also known as antidiuretic hormone. are important for, and aldosterone are important for salt, sodium, and potassium, and water control of your body. The structure of vasopressin is a peptide, a very small protein. The cells that release vasopressin are in the posterior pituitary. The stimulus that causes those hypothalamic neurons to release vasopressin in the posterior pituitary are an increase in the osmolarity of your plasma. So if your body is getting dehydrated, that is to say low water amount, so that your osmolarity of your extracellular fluid is going up due to dehydration, or the volume of your plasma is going down due to dehydration, that's what stimulates the hypothalamus to release vasopressin. The target cells for vasopressin are in the kidney, where they activate GS and GQ receptors that increase the reabsorption of water by the kidney. So if you're dehydrated, you do not want to produce urine to excrete water. You want to conserve water, and that's an increase in water reabsorption. If you want to know about the mechanism, take AMP2. It causes vasoconstriction, which causes an increase in blood pressure and an increase in blood volume. Again, if you want to know how vasopressin causes vasoconstriction, then take AMP2. And vasopressin also causes ACTH and TSH release to stimulate the corticotrophs, to stimulate the adrenal gland to make cortisol, and to stimulate the thyroid hormone to produce thyroid in a response to this stress of dehydration. Aldosterone, which is also critical for and required for life, for water and salt conservation. Aldosterone is a steroid hormone. Its stimulus is from the kidney. So if you want to learn more about this stimulus, take AMP2. The cells that release aldosterone, a steroid hormone, are in the adrenal cortex. So remember, there was a layer of cells there that produced both cortisol and aldosterone. Now we're talking about aldosterone. The target cells for aldosterone are also in the kidney. So they cause an increase in sodium, that is to say salt reabsorption in the kidney, to try to maintain a normal extracellular sodium concentration. The mechanisms by which aldosterone increases sodium reabsorption in the kidney are covered in ANP2. The receptors for aldosterone are intracellular, just like the receptors for cortisol, because the steroid hormone is amphipathic enough to diffuse through the cell membranes of the kidney cells and increase transcription of specific genes, leading to increased sodium reabsorption. So the vasopressin from the posterior pituitary and aldosterone from the adrenal cortex work together to conserve water and salt and maintain a normal salt and water balance in your body. Let's now turn our attention to calcium. We've talked a lot about calcium in this course. Calcium's required for neurotransmission. It's required for muscle contraction. It's required for bone growth. So we've talked a lot about calcium, and now we have to talk about how your body controls serum calcium levels in order to give you enough ionized calcium so that calcium can flow in through voltage-gated calcium channels, causing neurotransmitter release or hormone release or other forms of regulated exocytosis. And we need ionized calcium in the extracellular fluid to control that. We need calcium in the SR to allow for muscle contraction. So one of the hormones that controls calcium levels in the blood, in the serum, is parathyroid hormone. It is a small peptide produced by the parathyroid gland. Para means around, so parathyroid these are cells. Parathyroid cells are cells that are sort of in the thyroid gland, but are not follicle cells of the thyroid. So these are separate cells located around the follicle cells of the thyroid. They're kind of embedded in the thyroid gland, but they are not thyroid cells. They're parathyroid cells. The target cells for parathyroid hormone are kidney to conserve calcium, bone to to provide serum calcium if necessary from bone, and intestine to absorb calcium from diet. So we can either conserve it with the kidney, we can get it from our diet, or we can get it from our bone as sort of a last resort. The bone is a rich source of calcium. It's mineralized calcium. It's not ionized calcium. But we need ionized calcium to live. And so we'll take it from the bone if necessary. The receptors on those cells are GPCRs. And so what do they do? What does the activation of those GPCRs do in those organs? It increases calcium reabsorption. So the kidney, instead of excreting calcium, will conserve it dramatically and keep it in our body. So we don't want to excrete it. We want to reabsorb it. And the mechanisms are covered in AMP2. We will increase the absorption of calcium from our diet as much as possible. Let's get all the calcium we can get from the food we're eating in order to maintain serum calcium. And if necessary, we will mobilize calcium from bone. That is to say, we will stimulate the osteoclasts to break down bone in order to provide enough ionized serum calcium to survive. Those are the things that parathyroid hormone does to increase serum calcium if it goes low. Calcitonin is the antagonist. So if your serum calcium is high, then calcitonin tends to help reduce it back into a normal, healthy range. So calcitonin is a peptide hormone. It is also produced by the parafollicular glands. No, it's not produced by the parathyroid cells. It's produced by parafollicular cells of the thyroid itself. So there are other cells in the thyroid, not follicle cells, but parafollicular cells that produce calcitonin. So just to be clear, these are supposed to illustrate the parathyroid producing cells, and in the thyroid itself, there are parafollicular cells that produce calcitonin. The targets for calcitonin are just like the targets for parathyroid hormone: kidney, bone, and intestine. The GPCRs are GS and GQ linked. But when they are activated, calcium mobilization from bone is decreased, and in fact, calcium mineralization of bone is increased. So the excess calcium is put into bone. Absorption of calcium from the intestine is decreased. If we have a high level of calcium, we do not need to get it from our diet, and so we decrease calcium absorption and we excrete it in feces. Calcitonin also increases phosphate levels by increasing phosphate reabsorption from bone, and that's because calcium phosphate is one of the minerals in bone, and so having high phosphate helps put the calcium that is in excess into the bone. Now, you probably already know that calcium and bone health depends on another hormone, really, called vitamin D. Vitamin D is important for calcium and bone health. Vitamin D is a steroid that we synthesize in skin in response to ultraviolet light. It's now supplemented into milk. So if you look at your container of milk, it'll say something about vitamin D supplemented. So you can get vitamin D from milk and other dairy products, but you don't need to get it from milk because if you get enough sunlight, your skin will produce it. The targets for vitamin D are in the intestine, the bone, and kidney again. It is an intracellular receptor, or it reacts with an intracellular receptor because it's a steroid hormone. And vitamin D assists in the control of serum calcium, and it assists in bone production. So vitamin D and calcitonin work together to maintain bone strength and bone health. Well, so we've heard about antagonistic controls of calcium. We have a very narrow set point for serum calcium, 50 micrograms per mil, which is also 50 milligrams per deciliter. A narrow normal range of serum calcium is healthy. So what hormones control serum calcium? What goes in here and what goes in here? What hormone is stimulated by low serum calcium and would tend to bring serum calcium up? What hormone is stimulated by high serum calcium and tends to bring serum calcium down? Here's the answers. Parathyroid hormone is stimulated by low serum calcium, and it tends to increase reabsorption from kidney, increase absorption from intestine, and increase absorption from bone to increase serum calcium. Calcitonin is stimulated by high serum calcium. High serum calcium stimulates the production of calcitonin. Calcitonin decreases calcium absorption in the intestine. It decreases calcium reabsorption in the kidney, so you excrete it, and it decreases calcium mobilization from bone. In fact, it allows or increases calcium mineralization of bone along with vitamin D. So that's the antagonistic control of a essential ion in our serum, calcium ion.